and it's wonderful to be at NCCU, which I'm learning so much about, and it's such an impressive campus and place. So, as you just heard, my name's Molly Fox, and I'm going to be talking about Alzheimer's disease from an evolutionary perspective. So this talk might be a little bit different than the kind of more typical biomedical research talks that you hear. I've tried to make it fun because I know it's the morning. Before we get started, I just am curious how many people in this room have, say, some familiarity with the concept of evolution by natural selection, maybe like a college level course, if we could just raise hands. Okay, so absolutely like a large percentage. Okay, cool. So the field I come from is called biological anthropology. It's basically the study of human evolution. So people in biological anthropology, like myself, we study all different aspects of humanity, but united by this interest in human evolution. So we study human biology, human culture, human behavior, all of these different things, fossils, all united by this evolutionary perspective, which I feel is quite a powerful perspective because it helps us all see and embrace our common humanity. So it connects us all to a shared history, a shared story. We are all part of one family as humans, and we all share a very deep history together. It's also a powerful perspective because it connects us to the much larger story of evolving life on this planet. So I want to focus our attention a little bit now on mammals and think about what it means to be a mother if you are a mammal. So for a typical mammal mom, you have your baby, and then you take care of your baby, you lactate, you breastfeed the baby, and you are solely responsible for this baby, and as it grows and grows, eventually you wean it, at which point it immediately goes through maturation and becomes an independent adult itself. And maybe if you have cats or dogs, you're thinking, well, sometimes you have a litter. Yes, that's true. Sometimes you don't just have one baby, you have multiple babies, but they're all going through this pattern together. You're lactating all of them together, they're all the same age, they're all growing at the same rate, ostensibly, and then they all go through maturation, and then you can move on to your next litter or your next individual offspring. So this is a really different story than what we see in humans. Human mothers have a great challenge that no other animal faces, no other animal. Because after a human mother weans her offspring, it remains dependent on her. So anyone who has been a child or has had children knows that after you wean your kid, they're not going off and doing their own thing, right? You are still responsible for that child as a parent. You're still taking care of that child. And so ultimately, a human mother ends up having these children, multiple children of all different ages, so who have all different developmental needs. Just think about the different foods that you need to provide. So maybe you're breastfeeding a new infant and you're chasing after a toddler and you're trying to help your teenage child get over their first heartbreak. It's almost an impossible task to do all of these different parenting activities while also keeping yourself alive. But we don't do it alone, and that is our evolutionary adaptation as humans. We practice a kind of reproductive strategy that in ecology we refer to as cooperative breeding. Humans raise children in a more collective way than that panda mom I showed you on the first slide. We have a whole cast of characters who participate in various ways in supporting mothers in this kind of impossible task, allowing us to evolve the adaptation of having these overlapping periods of offspring dependence. And so it really truly takes a village to raise a human child. Now, we're not the only animals that have helpers in this way. Some other animals do. They don't have overlapping periods of dependency, but they do have other individuals participate in supporting mothers in the process of child rearing. It's somewhat rare in mammals. It's very rare in primates. But it's actually quite common among birds. It's so common among birds that we refer to this as having helpers at the nest. So even when we talk about ourselves, we actually often use this bird term because it's such a common reproductive strategy in birds that you have helpers at the nest. So you might have more in common with a bird than with a panda than you thought before this morning. Okay, so none of this reproductive strategy for humans would be possible without the help of these helpers at the nest. We refer to them as allomothers. Allomothers means anyone besides the biological mother who's participating in child care. Now, among humans, there's a whole bunch of different types of people who might help out in this process. Older siblings, aunts and uncles, friends, family, community, fathers, of course. But there's one class of individuals I really want to draw your attention to, and that is grandmothers. So across all human cultures that have been characterized in this way, grandmothers participate as dedicated allomothers. And they're important in this role because, unlike maybe the baby's father, they actually have a lot of experience already with child care. And so they can apply the knowledge that they have already acquired through their lifespan to help new parents. And also, from a sort of evolutionary ecological perspective, it's important to recognize that grandmothers, for the most part, are postmenopausal. This is also a kind of important feature of the human life story, which is that grandmothers don't have the competing interest of saying, maybe I'll help you with your baby, but I also need to go off and find a mate and have a baby myself, and maybe I'm taking care of my baby, because grandmothers aren't doing that anymore. So they turn their focus towards allocare. So with the help of allomothers, and especially grandmothers, human mothers are able to sustain this really fast pace of reproduction compared to other animals. When we think about ourselves as humans and compare ourselves to our closest living relatives, the other great apes, they have 8 or 9 years between offspring. But we don't have to wait 8 or 9 years, because when we still have our dependent toddlers, grandmothers and other allomothers step in and help us out, and this has led to the evolution of fast reproductive rate in humans. And importantly, this story has been applied to understand the evolution of why we live so long. Humans have, of course, a remarkably long lifespan that even extends beyond women's ability to reproduce. From an evolutionary perspective, this is a very unique trait to have this sterile period of the lifespan that evolution has promoted, potentially because grandmothers are able to increase their number of descendants, that's the currency of natural selection, number of descendants, by investing in grandchildren. So grandmothers are able to increase the number of descendants by investing in the number and survival and development and health of grandchildren, which leads to selection for long lifespans. So what you may be thinking now that I have raised this issue is this question: across human history, did grandmothers actually live long enough to play this role that I have just described? So we really have to address this question of how old is old age? When we look across human evolutionary history, how long have we been living into what we today consider to be old age? And I want to bring this up because it's a really common misconception that before modern Western medicine, people just died out in their 30s and 40s, and that was the typical length of the lifespan. Before we get into that, I just also need to explain what I mean when I say human evolutionary history. All right, this is a 10-week course in 10 seconds. The history of Homo sapiens, our species. So people just like me and all of you and all people that you've ever heard of and known first emerged around 190,000 years ago. So this is people who are in every way, morphologically, physiologically, cognitively, spiritually, the same as any other people. These are modern Homo sapiens, modern people. And humans lived exclusively on the African savanna and expanded throughout other parts of Africa, living as hunter-gatherers for about 130,000 years, at which point a small group of people migrated out of Africa, migrated around eventually all parts of the globe, and colonized virtually every habitat on Earth. It was only 8,000 years ago that we started farming. So at that point, we've been living for quite a long time, only as hunter-gatherers, right? And then 200 years ago, which is the blink of an eye in this evolutionary story, we completely transformed almost everything about our lives with the Industrial Revolution that began first in England and eventually reached almost every part of the planet. So with the Industrial Revolution, we completely transformed our environments, our social structure, our everything that you can think of. But the thing I want to emphasize is that that is not the environment in which we spent most of our evolutionary history. The vast, vast majority, 96% of our evolutionary history, was spent just living as hunter-gatherers. And so that's why I often refer back to that period, because it is the period and the lifestyle and environment that has shaped us the most. So here are pictures of 3 individuals who come from communities who today still live as hunter-gatherers. So these are not ancient pictures, these are contemporary pictures. These are photographs from people who continue to practice this lifestyle. So as anthropologists, we often study these communities, not because they're perfect replicas of the past. They're not. There are a lot of differences between them and their own ancestors and all of our collective ancestors, of course. But their lifestyles and environments much better resemble those of our collective past. And so we can learn a little bit, especially in this case, about human lifespan and human health, because they don't practice Western medicine, and they live outdoors, and they have a lot of other things much more in common with our collective past environments and lifestyles. So these individuals come from the Kung San in the Kalahari Desert, the Hadza in Tanzania, and the Aceh in Paraguay, as just 3 examples. And these are all pictures of older women. So as you probably were expecting, the life expectancy at birth in these 3 communities is just what I said it's not. It's about 30 to 40 years. The reason that this is not the shape of the lifespan is because life expectancy at birth is not a very good statistical indicator of how long people live. Because in these communities, infant mortality rates are so high. About 20% of babies don't survive their first year of life. And so if you imagine numerically, you're taking an average of the age at which everyone in the community dies, but you have tons and tons of individuals, 20%, who are dying at age 0. So it's dragging down that average so much. And so if you really want to know how common was living into what we today consider to be old age, what you might want to do is look at people who survived those first few early years and say, how long do they live? So there was a study of this question, and what they found was that 4 individuals in hunter-gatherer groups today who survived to age 16, so who basically survived to adulthood, the most common age at adult death was 72 years. And about 1/4 to 1/3 of all deaths occurred after that age. So living into our 70s is actually probably not a very modern phenomenon. It's probably something we believe from both this evidence as well as fossil evidence emerged earlier than Homo sapiens. This is a trait that has been around for as long as humans have been around. So why do we live so long? Well, I already talked to you about this grandmother hypothesis, this idea that grandmothers can invest in their number of grandchildren and increase the pace at which their daughters and daughters-in-law reproduce, and that can lead to the evolution of long lifespans. But I want to also point out something that even my colleagues in anthropology, we often overlook, which is that grandmothers can increase their number of descendants not just by participating in hands-on childcare, but also in participating in other activities like engaging in domestic chores that support the family and free up the parents to take care of the kids. So a kind of indirect way to achieve the same thing. Grandmothers can also collect and process food for the family. And in fact, across all different hunter-gatherer groups that have been studied, the life phase after age 50 is the only part of a woman's lifespan when she regularly produces more food than she consumes. Grandmothers also can participate in the labor and the activities of day-to-day life and teach culture and wisdom and traditions and strategies and information that they have collected over a long lifespan. And that can help their children and grandchildren engage and succeed in the challenges that they face in life. Okay, so grandmothers or older women are able to actually increase their what we call inclusive fitness, which is their number of descendants, by participating in all these different activities: childcare, food production, skills training, domestic labor, etc. All of these are activities that require intact cognitive ability. And so that is why Alzheimer's disease and cognitive health in older age more generally is an important question from an evolutionary perspective. We have to, I think, discern what cognitive health might have been across human evolutionary history, across that past that I just described, in order to understand why we evolved such long lifespans, why we evolved these overlapping periods of dependency, why we evolved all of the features that make the human lifespan and life cycle interesting and unique in what it is. So this is why I began studying Alzheimer's disease from an evolutionary perspective. Alzheimer's disease completely undermines the interdependent nature of human families. Our family social structure, like I said, it's a highly interdependent system. Alzheimer's, of course, also destroys our ability to think and to remember and to communicate. These are all the most fundamental features of our species. These are topics that I've written about before, which I expand on more in writing than I'm able to here today. But basically, the approach that I want to take now, this is one of the sort of ideas that comes out of evolutionary medicine, this is the subfield I'm in, is to try to figure out what was Alzheimer's disease risk in the pre-modern past. How are we going to do that? We don't have a time machine. This is a really hard thing. You can't really look at fossils and figure out whether someone had Alzheimer's. So what do? The best I've come up with is to say, well, what are the risk factors that modulate people's risk today? And then we can ask ourselves, were those risk factors any different in the pre-modern past compared to today? And that can help us try to draw some parallels and understand, would the same person living today and in the pre-modern past been at higher or lower risk for the disease? Now, when we go down this road, there are actually some risk factors that I feel like are so obviously different today that I want to bring them up front, even though these aren't even the ones that I study. And these are cardiovascular and metabolic risk factors for Alzheimer's. As many of you probably are quite aware, Alzheimer's disease is at much higher risk if you have the conditions that are listed here. Things like type 2 diabetes is a contributing risk factor for Alzheimer's, high blood pressure. And these are all conditions that we are quite comfortable saying were quite different in the pre-modern past compared to today. In fact, these are conditions that have been somewhat extensively studied in non-industrialized populations today, and they are virtually absent. Not completely. I'm not saying zero people who live today as hunter-gatherers have cardiovascular disease. I'm not saying zero people are obese, but it's very, very rare. And of course, we know these are quite common in the contemporary industrialized world. And that's because since industrialization, we live much more sedentary lives, we eat processed foods, and these are the major, major risk factors for cardiovascular disease and metabolic disease. So we can already infer some possibility that these risk factors would have been at lower prevalence in the past compared to today, and therefore the same person at the same age living in the pre-modern past compared to today may have had lower risk of Alzheimer's. But I want to share with you the two areas that I work in, which are not cardiovascular and metabolic disease. The first one is microbial diversity. Microbial diversity, it refers to the microbiome. So that's the microorganisms that live in and on our bodies. This is a major area of evolutionary mismatch. The microbes themselves that have lived in and on us were mostly the same across really all of mammalian evolution until quite recently when we industrialized. So with the Industrial Revolution, One of the changes that we made to our environments is that we started living in really densely packed, lots of people together kinds of environments indoors. Before this, we were living outdoors, and we were living in more sparse sort of populations. So this is an amazing opportunity if you are a bacteria or a virus or other kinds of pathogenic microbes, because this means that you can jump from host to host really easily. You don't even need your host to survive for more than 5 seconds for you to be able to jump from them to another person, to another person, to another person, and that leads to the evolution of virulence. That means that our pathogens became very, very dangerous, very aggressive. And this is where we saw absolutely devastating epidemics of infectious disease, like influenza and typhus and typhoid and cholera. And these kinds of conditions could never have existed if we didn't start living in these densely packed indoor environments. And so what did we do? As humans, we said, uh-oh, this is scary. So we invented ways to save ourselves, which was public health sanitation. We implemented public health infrastructure that we are very happy to say wiped out a lot of these pathogens. You're probably not walking around saying, gosh, I'm so worried I'm going to get cholera today. And that's because we've taken some measures to protect ourselves. This was an important and necessary step. We were suffering quite a lot until we did this. But in wiping out the pathogenic microbes, we also wiped out the benign, friendly microbes that at that time we didn't even really know existed. And again, I'm not saying don't wash your hands, don't spray your counters, don't drink sanitized water, don't use toilets. I'm saying all those things are great, and they're important and they're necessary, but everything comes at a cost. And there was a trade-off in this decision that we made, and this was a new problem that we created with the solution to the previous problem. And that new problem is that by wiping out those benign, friendly microbes, we harmed or reduced we impaired the development of our immune systems. Because benign, friendly microbes are necessary to educate the immune system. We're not born with a fully functional immune system. Our immune system requires input or stimulation from benign microbes during critical periods of development. And one of those critical periods is early life, like when you're a baby and a little kid. So our immune system is a system of checks and balances. Most of the cells in your immune system promote inflammation, attack foreign invaders, but there are some cells that actually suppress inflammation. They're regulatory, we call them. They basically control inflammation, right, like a natural Tylenol that our body makes. One of the most important cell types kind of comes about in the following way. We have these naive T cells, and they don't really do anything. And depending on what kinds of biochemical cues they're exposed to, they can differentiate then into all different kinds of subtypes. All of these subtypes in various ways promote inflammation or attack, except for one, which is the regulatory T cells. So regulatory T cells are the other side of that scale. They suppress or regulate inflammation. And what is the stimulation that causes a naive T cell to differentiate into a regulatory T cell? The origin of that stimulation is actually benign, friendly microbes. So in the absence of these, in the absence of a healthy, diverse microbiome, our immune system can get out of balance. And this can lead to inflammatory diseases or conditions like atopies, allergy, asthma, autoimmune disease, and importantly, Alzheimer's disease as well. Potentially falls under this category. Because Alzheimer's disease is a systemic disease. It doesn't just exist in the brain. There are biomarkers of Alzheimer's disease throughout the periphery as well, throughout the rest of the body. And Alzheimer's patients exhibit fewer regulatory T cells than healthy controls. So fewer of those kinds of T cells that suppress inflammation. And importantly, this difference between those who do and do not have Alzheimer's disease actually manifests pretty early in the lifespan before Alzheimer's disease symptom progression occurs. So this is something that we see in midlife. There are no changes in the ratio between different T-cell subtype compartments across the progression of Alzheimer's disease. So these differences we see between Alzheimer's and non-Alzheimer's patients manifests earlier. So I was interested in this phenomenon and thought that it could be possible that some of these differences between who does and does not go on to develop Alzheimer's could be due to different degrees of sanitization in the environments. Basically, environments in which we have wiped out more microbes, again, for good reason, but this might be one of the costs, may manifest in populations that go on to have higher rates of Alzheimer's disease than others. And indeed, this is what we found, that older people in countries that have more of these harmless microbes in their environment have lower risk of Alzheimer's later in life. And the converse, older people who lived in countries with more sanitized environments had higher risk of Alzheimer's. This was after a lot of control variables thrown into the equation. Along with a couple of graduate students in my lab, Delaney and Casey, we looked at how various microbiota in all different parts of the body can affect Alzheimer's disease risk. Why are we as evolutionary biologists interested in this question? Again, it's because the microbes that live in and on us, our microbiome, have changed so dramatically across evolutionary history. So I already mentioned how microbes participate in educating the immune system and differentiating regulatory T cells and thereby enacting better immunoregulation. Microbes also participate in all different ways, and I'm not just talking about the microbes in our gut. I know we're all really interested in those because that's where the vast majority of our microbiome resides, but there's also microbiome in your mouth and in your nose and on your skin and in your lungs. And microbes can contribute to inflammation depending on the sort of balance of benign versus pathogenic characterization of those microbiota, and can contribute to barrier permeability. So that's barriers for all different epithelial barriers in the body, actually. Barriers in the gut that allow things to translocate into the bloodstream that maybe you don't want there, as well as blood-brain barrier permeability itself, which can eventually even lead to microbial translocation into the brain, which has been in various labs are looking at this, associated with Alzheimer's disease pathogenesis, or just the translocation of pro-inflammatory cytokines that are permitted to cross the blood-brain barrier and contribute to neuroinflammation, which of course is a major component of Alzheimer's idiopathogenesis. In this project, we looked at a lot of different things. Beta amyloid is also a major player here because itself has antimicrobial properties, and there's a whole other area of evolutionary research looking at the antimicrobial role of beta amyloid and what that means for evolution of Alzheimer's, which we can get into in the Q&A. But I really want to get to the next part of our talk, so just to sort of put a cap on the microbial exposure side of things. This is something that has changed a lot across human evolutionary history, especially just in the last 200 or so years, in ways that ultimately make our bodies more pro-inflammatory, less immunoregulated, and can thereby contribute to Alzheimer's-related immunopathogenesis. So the other area that I'm interested in that has changed so fundamentally across human evolutionary history is what we call reproductive life history. So reproductive life history is the shape of the reproductive lifespan. That's things like when you go through puberty, how many babies you have, how far apart those babies are, when you go through menopause. These are all elements of the reproductive life history. And the two elements that I focus on are pregnancy and breastfeeding. So why are evolutionary anthropologists interested in women's reproductive life history? It's because women in the past had really, really different reproductive patterns than women do today. And these differences accumulate across your lifespan. An older person brings with them into the older phase of life the cumulative effect of a lifetime of biological functions. And the lifetime of biological functions varies so much between women in the past and women today. And the reason that I became interested in this is because natural selection for postmenopausal longevity, so living beyond menopause, occurred in the context of women's bodies that just were very fundamentally different than women's bodies are today because of the cumulative nature of this totally different life history. So just to take some examples here, when I write foragers, I'm referring to people who live today, contemporary hunter-gatherers. Agriculturalists refers to people who live today as small-scale subsistence farmers, and then typical post-industrial populations such as anyone living in North Carolina or Los Angeles, or et cetera. So these are just some population averages across multi-populations. The differences are so profound. For instance, the average age at menarche, so that's when women get their first period, among hunter-gatherers is 17 compared to the post-industrial women where it's around 12. The total number of times you ovulate in your life as a woman among hunter-gatherers is on average 160. You spend a lot of your life either pregnant or intensively lactating, and you go through menarche much later. Compare this to a post-industrial woman who's not on the pill, on average will ovulate 450 times in a life cycle. You can just imagine this is starting to add up to a very different biological experience. And then we get to the two areas that I'm most interested in, which are pregnancies. So I'm sure it comes as no surprise that in non-contraceptive populations, women have more pregnancies, 5.4 compared to 1.7. And this is something that you may not know, which is the length of breastfeeding actually until very recently would have typically been much longer. So this is measured in months per child. So and it varies a lot between societies, but the average duration of breastfeeding each individual child is about 2.5 years in non-industrialized societies. And now multiply that by how many more babies they're having. Women are spending so much more time, more than a decade of their lives on average, breastfeeding. And when you're breastfeeding, you have a completely different biological experience, and your body functions in a very specific and unique way that over a lifespan could contribute to a very different physiology that you bring to health in older ages. So from a purely biomedical perspective, it's also an important question how women's reproductive life history patterns relate to Alzheimer's disease. First of all, because we know that there are sex differences in the age-adjusted prevalence of Alzheimer's that are not these sex differences in incident Alzheimer's disease are not entirely explained by differences in longevity between men and women. And in fact, being a woman is the second largest risk factor for Alzheimer's disease after age. And layered on top of this, there's some very interesting evidence that there actually are sex-specific biological mechanisms. This is a paper I'm highlighting. It's a really interesting and important paper. I did not write it. I think it's really inspiring because it makes you think very creatively about Alzheimer's disease, because the same symptoms in men and women that we diagnosed nosologically actually manifest with different biomarkers in men and women and even different genetic risk factors. So this justifies the investigation of sex-specific processes in men and women. And of course, there's no more fundamental sex difference than reproductive physiology. So this leads to the sort of concept model that I'm working with here, which is the possibility that having more pregnancies, more breastfeeding, and a greater ratio of breastfeeding per pregnancy could enact alterations in women's physiology that are permanent and actually continue into their older ages and ultimately endow resilience against Alzheimer's disease. This is a sort of concept model that I've tested in 4 different studies, and I'll touch on each one a little bit here. So the first was a study of British women where I actually did Alzheimer's disease diagnosis. The second is a study of California women where we looked at cognitive function in older age. The third is a study using the pre-existing data set of the Women's Health Initiative memory study. This is a big national study that I'll talk about. And then the fourth is my current ongoing study, which is the Alzheimer's Life History Project. Okay, so in this first study, I recruited women who were age 70 to 100 years old, along with an informant, which was usually either an adult daughter or a spouse. This was a pilot study with 133 women. We conducted interviews and the clinical dementia rating scale. Okay, and as I show you the results, I don't want your eyes to glaze over, so I'm not going to talk too many numbers, but generally these are large effect sizes that were observed in this pilot study. We found that more cumulative time spent pregnant was associated with lower Alzheimer's disease risk. So what do I mean about cumulative time spent pregnant? This means that we are taking into account not just childbearing, but also miscarriages, spontaneous and elective abortions. So this was the first study that actually took into account all of the different kinds of pregnancies that women have, not just counting number of kids. And for some women, this really does dramatically change their amount of pregnancy that they've experienced. Because if there is a biological effect from pregnancy, it would be possible that that could have its effects regardless of whether the pregnancy lasted to term. So most people would interpret this kind of observation and relate it to estrogen. So there's a whole world of people who are interested in the long-term effects of estrogen in women's health. Now, estrogens rise exponentially over the course of pregnancy. The peak levels of estrogens during pregnancy are about 85 times higher than the peak during ovulation, so during the sort of other highest part of your lifespan. So, of course, you're getting a major dose of estrogen during pregnancy. Estrogen is really of huge interest to the Alzheimer's disease research community. It modulates brain networks. It inhibits processes that have been implicated in Alzheimer's disease. Almost anything that you can think of has been shown to be that estrogen has shown to have neuroprotective effects: beta amyloid formation, neuronal apoptosis, tau hyperphosphorylation, and many other processes as well. And we did indeed look directly at the amount of time women were exposed to estrogen in their lives, and we found a greater cumulative time exposed to estrogen. So that also takes into account hormone therapies during and after menopause, was itself associated with lower Alzheimer's disease risk. This was also a large effect size. Each additional month of estrogen exposure was associated with a 0.5% reduction in risk. In the second study that I mentioned, the study of California women, this was a slightly different study design. This was women age 50 and up. So the previous study we recruited starting at age 70. This was a similar study size. We did multi-domain cognitive testing, and this was actually a sample of women who were enrolled in clinical trials before the clinical trials started. So we basically took our data from the baseline visit. And this project was done in collaboration with a medical student, so I want to highlight her here, Hanadi. And we found, very similar to the British study, that greater cumulative estrogen exposure was associated with better performance on long-term verbal memory, which is, of course, implicated in Alzheimer's disease. So now to move on to some of the results related to breastfeeding. In the British study, we found that any versus no breastfeeding in your life and total time spent breastfeeding were both associated significantly with lower Alzheimer's disease risk. And again, these are really large effect sizes that we're seeing. For instance, any versus no breastfeeding, controlling for pregnancies, was associated with a 64% lower Alzheimer's disease risk. If I had a drug that could give you 64% lower risk of Alzheimer's disease, I would be so rich, it would be crazy. But and I wish I had that drug, but the observation that women's reproductive patterns have these kinds of profound effects on cognitive health and Alzheimer's disease risk, I think is very noteworthy. We also looked at the ratio of time spent breastfeeding compared to time spent pregnant. One of the reasons I'm very interested in this as its own construct, not just one controlling for the other, is because breastfeeding and pregnancy in many ways have opposite biological effects. So for instance, breastfeeding induces insulin resistance, pregnancy induces insulin resistance, and then breastfeeding restores insulin sensitivity. This is just one example, but the two sometimes balance each other out. So I think it's important to look at them together. So again, a higher ratio of time spent breastfeeding compared to time spent pregnant was also associated with lower Alzheimer's disease risk, also with a relatively large effect size. And similarly, in the California women's study where we did cognitive testing, we found that women who breastfed compared to those who didn't, and this is only women who had kids in this analysis, had higher scores in all of the cognitive domains tested, again with moderate effect sizes in this one. Okay, so the third study that I was going to mention is this analysis that I've been working on along with these wonderful collaborators using the Women's Health Initiative Memory Study dataset. So you may have heard of the WHI, the Women's Health Initiative study. It was this big clinical trial of hormone replacement therapy, mostly in the 1990s, and there was a sort of sub-study, the Women's Health Initiative Memory Study, which we call WIMS, and that's the data set that we are working with here. So this is a much larger study. The previous analyses I showed you had around 100 to 133 women in them. This is now 7,500 women. So as an anthropologist, I'm blown away. I'm sure all of you epidemiologists are yawning, but wow, 7,500 women, we can do so much. And there also was a sub-study of WIMS, so I guess a sub-study of WHI, called WISCA, the Women's Health Initiative Study of Cognitive Aging. So for 2,500 of these women, they had more extensive cognitive testing. So these women were enrolled in all of these various studies starting around age 65 on average, and they actually had annual cognitive testing in both studies. In WIMS, they did global cognitive function testing, and then in WISCA, they did domain-specific testing, and so I'm going to look at the memory testing scores. Okay, so, and these, sorry, I should say these analyses are not published, and I'm just sort of sharing with you the broad contours of the results. We found that women who had had pregnancies versus those who didn't, and again, more cumulative time spent pregnant, were both associated with better global cognitive function. Each additional month spent pregnant in your lifespan corresponded to 1% higher scores on cognitive function, every single additional month pregnant. Similar to the pilot studies, we also kind of corroborated those effects. We found that breastfeeding ever versus never and cumulative time spent breastfeeding was also associated with better global cognitive function, as well as better verbal and visual memory. And again, greater ratio of time spent breastfeeding to pregnant was also associated with better global cognitive function, verbal memory, and visual memory. So all of the results that we'd seen in the pilot studies, or at least for the most part, were endorsed. There seems to be a sort of convergence of effects, at least from my lab, that we are observing. So all of these studies sort of converge to suggest the possibility that women's later life cognitive health may have been better in the past compared to today, because as I was mentioning, women in the past had more pregnancies in a lifetime, more time spent breastfeeding, and a much higher ratio of breastfeeding to pregnancy, so much more breastfeeding per pregnancy. These, across these various studies, are shown to be associated with greater cognitive performance and greater Alzheimer's disease resilience, or lower Alzheimer's risk. So more cumulative time pregnant, again, we talked before that hunter-gatherers have much higher completed lifetime fertility compared to, say, American women. More cumulative time breastfeeding. This is, again, a really major and underappreciated way that women's physiology has that we've shifted our reproductive patterns in the modern era. That we can look both at fossil evidence, actually, for breastfeeding. This is one of the few things, because we can look at children's teeth and look at isotopes and see that actually among a hunter-gatherer group from about 12,000 years ago, breastfeeding per child was that average of 22 to 68 months, so that sort of average of 2.5 years that we see in contemporary hunter-gatherer populations seems to be pretty stable per child. So this all leads to the possibility that grandmothers in the pre-modern era may have been more capable of childcare and food production and domestic chores and skills training and all of these tasks that we have argued for so long among anthropologists have been the driving force of the evolution of human longevity and the driving force of the evolution of the human cooperative breeding system and the interdependent social structure of human families. And the two areas that I have sort of argued for this again are that we had probably more benign microbial exposure in early life, more pregnancies, more breastfeeding, and greater breastfeeding to pregnancy ratio. Now, I haven't established anything yet related to mechanisms. That's something that we're working on here in the Alzheimer's Life History Project. So this is a new study that we're doing in collaboration with my colleagues at the Pacific Neuroscience Institute in Santa Monica, where we are doing extensive characterization of women's reproductive life histories. We are doing our best to try to figure out what their early life microbial exposures may have been. This is all through, that's hard to do, but we're doing our best. We also look at early life psychosocial stress, which is another area of interest, and we try to look in this study at biomarkers. So we're actually trying to figure out what are the mechanisms that could possibly connect, especially women's reproductive histories, with their later life cognitive function. Why are these two things connected? So I'm going to give you a little bit of speculation about this. And this is, my own arguments of possibilities, but we haven't yet established any of these. The first is that we already talked a bit about immunoregulation. Well, I mentioned that early life is this critical period of regulatory T cell differentiation, but it's not the only one for women. Women get a second boost, and that occurs during pregnancy. In fact, the expansion of the regulatory T cell subset is necessary to sustain pregnancy. Levels of regulatory T cells increase during pregnancy, and actually only one study has looked at this yet, but continue to increase postpartum. And women who fail to expand this regulatory T cell compartment have recurrent miscarriage, high rates of spontaneous miscarriage, and infertility. Now, what we think is possible is that these higher concentrations of regulatory T cells would actually be retained in women's bodies for the rest of their lives, because during a pregnancy there's actually cellular exchange between a mother and a baby, and the baby leaves cells behind in the mother's body that have been shown to persist for decades. And so it's possible that the woman, that the immunoregulatory balance actually needs to be maintained with a higher concentration of regulatory T cells in order to tolerate these half-foreign cells that now live in her body forever. And estrogen also participates in this process. So these systems are all connected. The endocrine and immunological aspects of pregnancy are connected. So for breastfeeding, there are several neuroprotective biological effects that I think are possible. I already mentioned that during breastfeeding, insulin sensitivity is improved, and glucose homeostasis is improved, and also lipid metabolism is also enhanced by breastfeeding. And for these reasons, we already see for other diseases, decades after breastfeeding has finished, women who do breastfeed go on to have lower risk of metabolic disorders like type 2 diabetes, as well as hyperlipidemia. Now, there's a whole other way of thinking about this, which is the possibility that having more pregnancies and more breastfeeding actually has more of a social or psychological benefit that could be protective against Alzheimer's risk. because parenting and grandparenting enhance your social engagement and your mental stimulation and your emotional resilience. And so these are all ways that we probably are stimulating our cognitive function, building possibility of cognitive reserve for those interested in that idea. Having more adult children may just decrease your levels of loneliness in older age. These are more sort of simple explanations than thinking about, T-cell subtypes. But these are also all things that we associated with cognitive health in older age. And so all of these different kinds of mechanisms are what my lab is trying to kind of figure out right now, how these all play out. So when we look at the public health impacts here, I think that it's important to recognize that reproductive patterns around the world are changing. I'm not trying to be Elon Musk here and say that it's good or bad, that people are having more or fewer kids. I'm just saying that it's happening, and that women are having fewer and fewer pregnancies, fewer and fewer babies than ever before. And as the global fertility rates declines at such a rapid pace, we have to be prepared for what this means for the future of Alzheimer's disease, and actually prepare ourselves for the possibility that we may be facing an even greater future epidemic of Alzheimer's than we have considered. So in sum, this biological anthropology perspective helps us to understand ourselves, understand the ways that our bodies have changed over time, the way that we're built. And I think that it can give us new ways to think about human health and disease using this evolutionary, cross-cultural, and long phase of history kind of perspective to help us not just understand ourselves better, but come up with new, interesting ways that we can care for each other, especially caring for each other across generations. So with that, I want to thank all of my collaborators, my funding sources, and all of you for listening. Thanks. Okay, I guess we have a couple minutes for questions. Yeah, go ahead.
Thank you. My question is, with all of the studies, or any one of the...
Check one.
Sorry. I can repeat your question for the first time.
Who's asking a question? Okay, great. Sorry. Thank you. Throughout the studies, you talked a lot about the, breastfeeding, and you talked about cognitive function, and the impacts of breastfeeding. But throughout any of the studies, was there any talk or any data collected about postpartum depression, since that does affect memory loss and cognitive function? Yeah, it's such a good question. Okay, so about postpartum depression, which is itself associated with, impaired memory and stuff. Yeah, so this is a really interesting area, actually. And so in these studies, no, we do not, unfortunately, have those data. However, it is true that generally, both during pregnancy and postpartum, women often experience what's sometimes termed mommy brain, whether or not they have postpartum depression, but that is itself, even an aggravating factor, that there could be some memory impairment. So in our studies, we don't observe a persisting memory deficit, but I do also think that in terms of these evolutionary mismatches, I think that postpartum depression, which I happen to also work in that area, is like another area of that's sort of ripe for the analysis of an evolutionary mismatch there, because with the sort of fracturing of the social structure around women during pregnancy and breastfeeding, it would be, of course, perfectly logical that that would be associated with enhanced risk of postpartum depression. We know that lack of social support is one of the major risk factors, and we sort of expect women in the post-industrial world to be completely isolated from their normal day-to-day lives and have none of the social support that we evolved with to go through the experience of late pregnancy and postpartum. So I do think that these things probably pile on to all contribute to just poor mental health for mothers in both the immediate and the long term. Thank you.
This is really fascinating. Thank you for your work. It's important.
I'm just curious if you can give an example of how you determine early life microbiome development exposure.
Yeah, so this, it's a great question, and it's something that's very hard to do. So in the first study that I mentioned, the 2013 study, to determine the early life microbial exposures, we used these historical indices of infectious disease, sort of assuming that that would correspond to the unmeasurable benign microbes. And we also looked at public health infrastructure itself, so the percent of the country that was using, for instance, like toilets and those kinds of things. So that study was using these like sort of broad epidemiological measures. I think we had like 6 different measures that we were then taking some z-score of or something, to a principal component actually in that study to sort of say, well, all these things together would kind of indicate this. And we specifically were looking at the period where women who were developing Alzheimer's, that was a 2002 data set, so in 2002 would have had their early lives. So we sort of like calculated back and looked at that period of history. For the new study, we're trying to think of new ways to do this. I have another study that's actually in pregnant women where we do something similar, and in that one we look at sort of like rural versus urban residents, interaction with outdoor animals, interaction with pets, and interaction with other kids. So we've even done interviews with like the size of your kindergarten class, or whether, you know, your birth order, whether there are other kids living in the home. So we try to get at it in all of these indirect ways, and we've been having a lot of conversations about trying to think of metabolites or interesting biomarkers that might be good indicators, as opposed to just looking at your current microbiome, but that has, you know, potentially shifted over the lifespan. Oh, maybe whoever had their hand up first. I think she did. Sorry.
Thank you. That was a wonderful talk, and I am very interested in ApoE, in part because I'm at Duke, so that's where it came from. But with respect to Alzheimer's, but additional studies have shown in South American indigenous tribes that fecundity is much higher with ApoE4. As well as lack of Alzheimer's in those populations. Mm-hmm. We've also collaborated with people in Brazil, showing that, in fact, early onset development with ApoE4 is much, much better in children, especially those that are exposed to high levels of bacteria and, you know, poor sanitation. So, not to take away from women being female is important for Alzheimer's, but how do you reconcile the ApoE4 traits, you know, with sepsis and all of this other stuff versus being a woman, and I guess being pregnant and having children is yet another variable that adds to the protection in a sense.
Yeah, I'm so glad you asked about this. It's such an interesting area. So I actually think that ApoE4 is another great example of this kind of, it contributes also to this evolutionary story in an important way, because I don't believe that in the pre-modern past, I don't believe that we have any evidence to think that ApoE4 would have contributed to greater Alzheimer's risk. The reason for that is that we have actually, all of these studies, like you're saying, not just the Chamane, because that's a midlife study, I think, right? They were in their 50s or 60s then. But there have been lots of studies. There was a study, anyway, there's been studies of the San people in the Kalahari. There's studies of small-scale agricultural village communities in the Middle East. There's a study of the Yoruba in Nigeria. And they all show that E4 is prevalent in these communities, but has no association with dementia risk or Alzheimer's disease risk. And the possibility, I think, is that if E4 is contributing to risk through some sort of inflammatory pathway in the absence of, which I know is an if, but in the absence of this sort of hyperinflammatory physiology, it wouldn't have that same contribution to risk. And a lot of evolutionary biologists are interested in this question of, well, if the E4 variant of ApoE increases Alzheimer's risk, then why do we still have it? Wouldn't natural selection get rid of it, especially because grandmothers are so important and all this? But I would argue that if it does not actually increase risk in a pre-modern context, then we don't really need to figure that out. E4 is the ancestral allele, so it's something quite similar to it, not exactly similar as what we see in all primates have E4. So it's actually the E2 and E3 variants that have evolved later, but the E4 variant has persisted. So I think that, and then, so then the other aspect is there is this possibility of pleiotropy, where in early life conditions like survival against various diarrheal diseases and infections, E4 actually appears to give some kind of advantage. So that also could argue a kind of different evolutionary explanation for the persistence of E4, that it could have a potent sort of advantage in early life that might counteract the later life disadvantage. But like I said, I argue that the later life disadvantage is even further diminished in sort of non-Western, non-industrialized contexts. I think the evolutionary story behind E4 kind of deserves a little bit more extrapolation in those ways.
Thank you.
Hi, thanks so much. Really thought-provoking and interesting talk. I'm curious to hear your perspective in terms of thinking about competing risk and survivorship bias and immortal time bias as sort of potential threats to the sort of causal relationship that you posit between a pregnancy, breastfeeding, or for that matter, early life microbiome. In particular, thinking about, from the really interesting lens you bring up about the longitudinal evolutionary history, recognizing that the competing risk of death we as humans face in a post-industrial society is dramatically different than the risks that our evolutionary ancestors faced, and how you think that potentially can generate future insights for how you sort of approach this kind of work.
I think that's a great question, and it's a really interesting question, very difficult question to address. But yeah, if we have, let's say, 20% of infants in a pre-modern medicine context or in a non-modern medicine context who don't survive infancy, then was there something sort of biased about that population, that somehow vulnerability, say, during the first year of life, somehow is, of course it would be non-biased survivorship. There'd be some vulnerabilities in some way, say susceptibility to diarrheal disease or something, which would be a common, cause of death in early life. And that could somehow bias the sample that survives to later be maybe in some other way resilient against Alzheimer's disease. I think it's an interesting question. It's not something that I've been able to establish in my data. Another thing that people have looked at is the effect of pregnancy itself on longevity. There are some studies that show that pregnancy could be associated with reduced longevity. We don't see that in the Women's Health Initiative cohort, but and other cohorts show some kind of a U-shaped probably curve. But I think that these are really interesting epidemiological questions that kind of, that we try to look at like in these various ways as best we can. But I definitely think there's a lot of opportunity for further trying to understand this. And a great way to think about it would be, are the risk factors for Alzheimer's disease similar to the more sort of traditional causes of death? Right now, I don't see a huge argument for that, only because these sort of like metabolic and cardiovascular risk factors, which I said are kind of straightforward, are not the same risk factors that we see as being very playing a major role in early life mortality in a more sort of traditional pre-modern setting. But yeah, I think it's a really interesting question.